Hey students, this is your Nandini ma'am, and today we are going to continue our audio chapter, The Cell Membranes Gateway to Cells. Let's get started. The detailed molecular structure of a cell membrane cannot be seen even with an electron microscope. Many years of research have led to the development of a number of different models that have attempted to explain the structure of the cell membrane. Our current accepted understanding is based on a model proposed by 1972 by Singer and Nicholson called the fluid mosaic model of cell membranes. This model, with minor modifications over the years, has been accepted because the behavior of membranes, estimated surface area, chemical analysis, and electron microscope studies are all compatible with the model. It also accounts for most functions associated with cell membrane. The fluid mosaic model. The cell membrane controls the exchange of material between the internal and external environments of the cell. It is selectively permeable, meaning that it allows only certain molecules or ions into or out of the cell. The structure of the cell membrane allows the concentration of substances inside cells to remain fairly constant and different from the external environment. In the currently accepted model, the structure of the selectively permeable cell membrane is described as a fluid mosaic. This fluid mosaic model proposes a lipid sea with many and various proteins floating on and in it. The fluid mosaic model describes the cell membrane as a double layer of lipids, a lipid bilayer with the ability to flow and change shape like a two-dimensional fluid. Specialized protein molecules are embedded in the lipid in various patterns like a mosaic. Some of these proteins can move sideways, but others are fixed in position. Both proteins and phospholipids help to control the exchange of materials between the external and internal environments. Lipid component. The fluid part of the cell membrane is composed of two layers of phospholipids forming a phospholipid bilayer. Each phospholipid in these layers can be represented by a head and two tails as shown in figure 2.31. A phosphate group on the head makes this end hydrophilic, able to absorb water or dissolve in water, otherwise known as water-loving. The fatty acid tails are hydrophobic, water-avoiding or unable to dissolve in water, or water-hating. When these molecules form a bilayer, the water-attracting heads are positioned facing outwards towards the cytoplasm on one side and to the outside of the cell on the other side. The water-repelling tails are positioned inwards towards each other. This layering is termed a bilayer. It is not rigid in structure, hence the term fluid mosaic. A type of lipid called cholesterol is interspersed among the phospholipid molecules in animal cells. This makes the membrane more flexible. Membrane flexibility in plants is increased by a different lipid, phytosterol. The lipid components of all membranes allow them to be flexible and repair themselves. This means that the cells can change shape and grow. Cell membranes are able to break and reassemble themselves during processes such as cell division. This structure forms the basis of the cell membrane and all other membranes within cells, such as those surrounding organelles. Proteins are then interspersed throughout this structure. Membrane proteins. Protein molecules are scattered throughout and suspended in the lipid bilayer. Some proteins penetrate all the way through the bilayer, forming channels that allow some materials to cross the membrane. Other proteins may be partly embedded in the membrane. It seems that some proteins are fixed in place while others travel about freely. So the proteins described as floating in the lipid bilayer, like icebergs in a lipid sea, giving a mosaic effect. Some proteins function as pores, temporary or permanent, or form active carrier systems or channels for transport, while other proteins have carbohydrates attached for cell recognition. These proteins enable cell-to-cell interaction and communication and the exchange of substances between the cell and the external environment. For example, in multicellular organisms, adhesion proteins link cells together and help maintain the organism's three-dimensional structure. Transport proteins act as passageways that allow specific substances to move across the membrane. Membrane proteins are also involved in cellular communication. Receptor proteins are different in different types of cells. They cause the cells to respond only to certain signals from substances such as hormones that bind to them, giving them specific functions. Membrane recognition proteins or glycoproteins are made up of a protein molecule with a carbohydrate molecule attached. These proteins identify the cell and are called antigens or marker molecules. They allow the immune system to distinguish between foreign particles and the body's own cells. This ensures that the immune system will destroy only foreign particles that display non self markers. Investigation 2.2, Modeling the structure and function of the fluid mosaic model. Models are used in science for a number of reasons, including to simplify a concept, make a visual representation of something that can't be seen. There are many ways of modeling the structure and function of a cell membrane. Aim to model the structure and function of the fluid mosaic model of the cell membrane. Materials. Structure. Rectangular cake or polystyrene block approximately 20 cm into 10 cm into 10 cm. Knife, icing sugar, food coloring or fondant or buttercream. Marshmallows, licorice or modeling chocolate, selection of lollies, labels or toothpicks. Function. 2 into tea strainers, icing sugar, lollies such as Smarties, elastic band, colored paper, salt, sugar granules, tea leaves. What are the hazards? What risk does the hazard pose? How can you safely manage this risk? Knife or scalpel. Sharp edges can cause cuts. Use knife scalpel with care and keep fingers away from sharp edge of knife. Traces of chemicals. Contamination of tabletops. Do not eat the cake if it was prepared in a science laboratory. Method. Structure. Trim cake to approximately the suggested size. Using fondant or icing, completely coat the surface of the cake. Place marshmallows to completely cover the top surface of the cake. Then place one line of marshmallows along the top and bottom of all sides of the cake, lined up with the marshmallows on the top edge of the cake. Using licorice or modeling chocolate, make tails. Stick 2 of these tails per marshmallow on the side of the cake leading from the marshmallow to the center. Using modeling chocolate or lollies, construct proteins that penetrate the whole cell membrane layer and proteins that are only partially embedded in the membrane. Attach lollies or modeling chocolate to some of these proteins to represent the glycoproteins. Use lollies or modeling chocolate to represent cholesterol and phytosterols. Attach labels to all parts of the model. Place 2 teaspoons of icing sugar and 2 teaspoons of lollies into each one of the tea strainers. Tie the handles of the tea strainers together with the elastic band. Shake the tea strainer over a piece of colored paper. Record in the table provided which substances pass through. Repeat this process using a variety of other substances. Results. Structure. Draw a diagram or take a photo of your model. Copy and complete table 2.3 to indicate what each part of your model represents in the fluid mosaic model. Function. Draw a diagram or take a photo of your model. Complete table 2.4 to indicate what each part of your model represents in the fluid mosaic model. Discussion. Structure. Outline the purpose of your model. What are the benefits of your model of the fluid mosaic model of the cell membrane? Discuss the limitations of your model. Justify the validity of your model. See page 4 for a discussion of validity. Function. Outline the purpose of your model. What are the benefits of your model of the fluid mosaic model of the cell membrane? Discuss the limitations of your model. Justify the validity of your model. See page 4 for a discussion of validity. Conclusion. Write a summary statement that relates to the aim of this investigation, including a brief summary of what you did and what you found. Key concepts. The cell membrane is selectively permeable and performs the function of controlling the movement of substances into and out of the cell. The currently accepted model of the structure of the cell membrane is the fluid mosaic model. This model depicts a fluid phospholipid bilayer with different types of proteins embedded in it, creating a mosaic effect. Proteins either penetrate from one side to the other or are only partially embedded. Some membrane proteins from pores, some form active carrier systems or channels for transport, and others have carbohydrates attached for cell recognition. Cholesterol or phytosterols provide some flexibility to the membrane. Models are used in science for a number of reasons, including to simplify a concept, make a visual representation of something that cannot be seen, or make predictions of expected results. Before a model is accepted, it needs to be validated. That is, certain predictions should be made, and when tested using the model should hold true. When modeling concept in the laboratory, all parts of the model should be related to actual parts of what is being modeled. The limitations of the model should also be documented. So, this was the chapter Cell Structure and Technologies. Happy reading.